Okay, maybe we could go ahead and get started. It's my great pleasure to introduce to you Dr. Moritz Stolla, who is the director of the platelet transfusion research activities at BloodWorks Northwest Research Institute in Seattle. He's also an assistant professor in the hematology division at the University of Washington Medical School. He is an assistant member at BloodWorks Northwest Research Institute, and he's also the associate medical director of the blood transfusion service at Swedish Medical Center. He's been doing some really fascinating work on cold stored platelets, and without further ado, I will turn it over to you, Moritz, and you can share your slides and enlighten us.
Okay, well, thank you very much for giving me the opportunity to present to this group. Hope you all can see my slides.
We can.
I'm going to talk, great, thanks. I'm going to talk about cold stored platelets, and as the title suggests, this is not really a new idea. This idea has been around for a long time, but the question is, should we really use them in the future as well? Yeah, just to give everybody up to on the same page about platelets and platelet transfusion, we currently can store platelets up to 7 days with additional bacterial testing. They are stored with agitation at room temperature, so 20 to 24 degrees centigrade. Most institutions in the US use plasma, but you can also use additive solutions, so Intersol and Isoplate, PAS-C and PAS-F. The dose is 3 × 10⁻11. You can get that from one apheresis donor, or you can pool whole blood derived platelets to get to the same dose. The volume is usually variable between 200 and 300 ml. And there are a couple of issues that need to be pointed out. This short shelf life, if you compare it, for example, to red cells, leads to frequent shortages, outdates, and wastage. And the supply is especially challenging in rural areas and far forward military scenarios. But even in major metropolitan areas like Seattle, we have routinely have platelet shortages. And sepsis from platelet units is actually the most common transfusion transmitted infection. So ultimately, if you look at all of these issues, it all comes down to room temperature storage, because it has been shown that after 5 to 7 days, the sepsis, septic events increase sharply. And that's why we have to reduce the shelf life to 5 to 7 days. And so how did we get to these current storage conditions? And this is a landmark study that was published in the late 60s by Scott Murphy's group, where they performed radiolabeling experiments in platelets that were stored at different temperatures. And you can see that even after only 18 hours in the cold, the circulation time was severely reduced for these platelets. And clearly this was not the case when they stored platelets at room temperature here. They also increased the temperature further to 30 and 37 degrees, but as you can see here, the yield, which is, I guess, the in vivo yield, so that recovery actually decreased with an increase in storage temperature. And there was no benefit in survival. So overall, and clearly, I mean, you can expect bacterial growth to be even stronger at 37 degrees. So ultimately, and with the patient population of hematology, oncology patients in mind that need platelets to circulate for a very long time, they concluded that room temperature is the way to go for these platelets. And if you look at the current transfusion landscape, though, this is from a relatively recent paper in Transfusion. They looked at the ICD-9 and 10 codes for platelet transfusions in academic centers. They found that only really 50% of all platelets go to patients with leukemia, MDS, and lymphoma. So platelets, patients that need platelets for prophylactic indications. And presumably the other 50% are much more likely to go to patients with active bleeding problems like trauma patients, surgery patients, and ICU patients. So what you could envision in the future is a dual inventory where you have room temperature stored platelets for prophylactic transfusions, so for patients that need a long circulation time, and you could reserve cold platelets for therapeutic transfusions where you the immediate function is much more important than the survival time. So what do we know about function of these platelets? There are some historic studies. I'm going to go over them just briefly and not too much detail, but this study here by Dr. Aster's group looked at the function of these platelets in aspirinated healthy volunteers, and they found that the cold platelets were better at correcting the bleeding time compared to room temperature stored platelets. But then they also showed that in a subsequent publication that the control group, so the room temperature stored group, was stored at a disadvantage. Similar in the same paper, actually, the thrombocytopenic patients received cold platelets compared to room temperature stored platelets. And again, they concluded that these cold platelets were more potent because the number of improved subjects was higher than in the cold group compared to the room temperature stored group. But the same limitation applies for this publication as well. Then my predecessor here at Bloodworks Northwest, Dr. Charles Schlichter, performed experiments also in thrombocytopenic patients. So this is the so-called hockey stick plot where you plot the platelet count, the bleeding time. These were patients with thrombocytopenia and platelet counts below 10,000. So they were all on the lower right corner here. And when she transfused fresh platelets back to them and measured the bleeding time, she was able to bring them back where you would expect them to be more or less in that area of where the hockey stick is. She then did the same experiment with room temperature stored platelets, and again was successful in correcting the bleeding time and increasing the platelet count. But then in the cold group, she was able to increase the platelet count, but the bleeding time did not decrease in these patients. Now, even though there are a lot of data points on these slides, I believe there are only 2 or 3 patients included in this study. So clearly, and the number is a limitation here. And Dr. Valeri's group also looked into platelet function depending on storage time, and he found that with fresh platelets, he needed 4 units to correct the bleeding time in healthy humans on aspirin. He needed 8 units for room temperature stored platelets and 1 unit for cold platelets, indicating that you have a decrease of function with the room temperature storage, but an increase of function with the cold group. But all of these studies have limitations. We use apheresis platelets now mostly. There's a storage time limitation with 1 to 3 days that were used exclusively for these studies. So given from what I've told you in the beginning that we can use room temperature stored platelets up to 7 days, this is really not helpful. Overall, the number of subjects included was very low. The bleeding time in and of itself has issues. It has a large intra and inter-operator variability. And as I mentioned, some retractions or corrections at least have been published subsequently for some of these studies. So when I joined Bloodworks Northwest, one of the first questions we try to answer are the effects of long-term storage on these platelets. And what we did was compare different storage times from 5, 10, 15, and 20 days in a regular platelet unit without agitation. We radio-labeled these units and traced in vivo recovery and survival. Now, I know this is not looking at function, but it was just meant to be a study to test on, I guess, what we can expect and how long they circulate. We also had a historic 5-day room temperature stored control group. And here you can see that the recovery of these platelets decreases with every storage time that we tested. So in 5-day storage steps, basically up to 20 days. So you saw a stepwise decrease in recovery. And for the survival, we kind of reached the bottom after 10 days. Now, this could be because we reached the limitation of the test. So the way to detect even a further reduction, we were not able to do that with the test, presumably. But if you look at these traces here, you can see that they're already pretty low in terms of recovery. Now, to address function, we use a flow chamber assay in collaboration with Dr. Nate Snydecki's group from the Department of Engineering at the University of Washington. We obtained fresh whole blood, depleted this whole blood, and then reconstituted platelets, either fresh or 5-day cold or 5-day room temperature stored platelets, and then perfused them through the chamber, which has collagen-coated block and posts. Now this, you can see this here. In this assay, you can detect the deflection of the post, and this is reflective of the clot retraction, which is actually working against the shear stress, and also the size of the aggregate can be assessed. Now, I tested these videos already, and they should work. So what you can see is that an aggregate forms. The deflection of the post you can actually not see with your eye, but it's detected by the system. And what we found is that there's a decrease in aggregates, but the cold platelets at least start out with a larger aggregate. Now this is when we the quantification of the data, and overall we did not detect any significant differences in force generation in any of these platelets. What was quite remarkable was the donor-to-donor variability here in all groups, and this is one of the questions that I think we still need to answer, why we have such a large donor-to-donor variability in our platelet donors. And here the area is shown below, and as you can see here, that cold platelets formed a significantly larger area compared to room temperature. And this is also true for the fresh group here. But in the later stages of the experiment, so after 300 seconds, we did not detect this significant difference anymore for the cold platelets. Now, to test platelet function after transfusion, we invited healthy volunteers and tested their platelet function at baseline to make sure they didn't have any endogenous bleeding disorder, platelet dysfunction. We then collected their platelets and stored them at either cold or room temperature for 5 days based on randomization. We then invited them back on day 4 and gave them a loading dose of aspirin and clopidogrel, and performed a platelet test to make sure that the endogenous platelet population was inhibited. We then performed the transfusion of the stored unit and tested their platelet function at 1 and 4 hours after transfusion. Then on the next day, we had a 24-hour time point, and this was then followed by daily phone calls to make sure that they were not bleeding. This same sequence was then done again with the alternative products. I mean, they're randomized to cold, they received room temperature in the second arm, and vice versa. And here's an overall outline of the study. Overall, we screened 28 subjects, 19 were eligible, and 14 were enrolled. We had one QC failure that was in the room temperature stored arm, and ultimately we're able to include 8 in the cold arm and 7 in the room temperature stored arm. And here's the sequence of transfusion. So most were randomized to receiving cold first and room temperature second. And this empty box is here because of the QC failure. And the number, there was a concern that the number would be significantly lower in the cold group, the number of platelets transfused, that is, but that was not the case. There was actually a trend for a slightly higher number in the cold group. Now, when we looked at the absolute platelet count, you can see that there were no significant difference at loading dose in 1 hour, but 4 and 24 hours, we saw a significantly lower platelet count in the cold group. And when we looked at the corrected count increment, so that takes into consideration the body surface area, the count increment, and the dose that was transfused, we also saw significant differences after 4 and 24 hours. And here are some of the aggregation data that we obtained. So this is at 1, 4, and 24 hours after transfusion. What was the most surprising finding for us was that we saw a significantly better response with room temperature stored platelet when we stimulated with collagen. This was here significant at 1 hour, but there was still a trend at 4 hours and to some degree at 24 hours. So arachidonic acid tests the aspirin pathway. And here we saw at 1 hour, basically equivalent reversal, but at 4 and 24 hours, you can see a decrease in the cold group. And with ADP, we did not see any significant reversal, unfortunately. So ADP is for the P12 arm, the clopidogrel inhibited arm. This is the Verify Now aspirin data. So this Verify Now assay uses whole blood. So we were not able to adjust the platelet count. The aggregation data here is obtained with washed platelets. So the differences in platelet count shouldn't matter for this assay. But in the whole blood assay, we were not able to correct the the platelet count. But nevertheless, so everything below that dashed line is considered inhibited. Everything above the dashed line is uninhibited. So both groups were, or both transfusion recipient groups were able to lift them back into the uninhibited area. But with the cold platelets, after 24 hours, they were back in the inhibited area. I think this is also good endogenous, like internal control, I guess, because the endogenous platelet production was not able to correct the aspirin inhibition on its own in the cold group. And here you can see the bleeding time assay. I mentioned already the bleeding time has numerous problems, intra and intra-operator variability. Overall, what was surprising is that with cold platelets, we saw them approximately 50% increase the bleeding time, 50% decrease the bleeding time with room temperature. The majority appear to correct the bleeding time. Now, collagen, I mentioned the difference between the response in cold and room temperature stored platelets. With collagen, the major receptor for collagen on platelets is GP6. Overall, we saw significantly lower GP6 levels with flow cytometry on cold platelets. Another collagen receptor is alpha-2 beta-1. Overall, we did not see significant differences here. To confirm this further, we performed a mass spec testing of platelets and were able to corroborate the flow cytometry findings that the cold platelets have significantly lower GP6 levels on their surface. Now, this is not, I mean, this assay involved humans that are on dual antiplatelet therapy. So we wanted to confirm this in an animal assay, just focusing on the transfused platelet population. And to do that, we used mice that express GFP ubiquitously. And these platelets were stored for 24 hours. We then transfused them into wild-type animals and performed blood draws at 4 and 24 hours, and were able to identify the transfused platelet population by focusing or gating on the GFP positive population, and then specifically looked for platelet activation in the GFP positive population. So very similar to the human situation, you can see that cold platelets have a shorter survival in mice. We could not use collagen for the flow assay because it's more difficult to stimulate platelets by flow with collagen. So we used convalescent, which is actually a snake venom, but specifically stimulates platelets with convalescent. And what we found is similar to the human data with collagen, that the cold platelets were less responsive to convalescent compared to room temperature storage. And we again looked also for the GP6 levels, and both of them were significantly lower in the cold group at 4 and 24 hours. So ultimately, this study is summarized in this little cartoon with the different GP6 levels leading to reduced response in collagen and convalescent in mice. Now, there was a study previously done by Denise Mark's group from Australia, where they looked at GP6 levels in stored platelets. And what she found, there was still a trend, I guess, for lower GP6 levels in cold platelets, but it was not significant. And these were actually platelets that were stored in additive solution. So we're currently looking into to also stimulating these platelets to see if that response to collagen is rescued, at least to a certain degree, after storing additive solution. And another encouraging study from Norway was published recently, where they looked for bleeding outcomes in cardiac surgery patients that received either room temperature stored platelets or cold platelets, 1 to 7 and 7 to 14 days. And here is shown the chest tube output, so a surrogate marker for bleeding. And overall, there were no significant differences detected here. Another way to look at bleeding in these patients is to look in to the blood component use. And again, there were no significant differences detected here. There was actually a trend for lower use in the cold group. But since it was a pilot trial, it was not really powered to show differences. But also in this trial, it's important to point out that they used additive solution for their cold platelets. Well, our studies were exclusively performed with 100% plasma. So, and the most recent study that we, I guess, are still working on, I'm going to show now. I mentioned that we used 5-day cold, 5-day room temperature storage in the last study. With the current study, we sort of replicated the same experiment, but using 7-day room temperature stored platelets and 14-day cold platelets. And because we didn't see any reversal with clopidogrel, with either group, we only used aspirin to inhibit the endogenous platelet population. And similarly, they were randomized to one arm in one round, and then after a washout period, they had a crossover and the repeated sequence with the alternative product. This time we were able to include 9 in each group. So overall, this is an overview of the study. Again, we had a QC failure in the room temperature stored group. But overall, 9 were included in each group, as I mentioned. This time we had a trend for a lower platelet count in the cold group, but it was not significant. Overall, no significant differences were observed in the platelet count. The TX here indicates the time of transfusion. Aspirin is after loading dose with aspirin, and the same time points here, 1, 4, and 24-hour post-transfusion time points. Here's the corrected count increment, no significant difference after 1 hour of transfusion, but 4 and 24 hours, similar to the previous study that I showed you, were significantly lower in the cold group. The Verify Now is the whole blood integrin-based platelet activation assay. Similar to the previous study, we saw a reversal with the room temperature stored group. With the 4-hour time point, we didn't have as many numbers included due to test failure, unfortunately. So I'm not really sure why that would go down and go up again, but for the 24-hour time point, we still had the same numbers or all numbers included. So again, with 24 hours, we see a significant reversal in the room temperature group, but really not much of a change in the cold group. Here are the results for collagen. After 7-day room temperature, 14-day cold, we didn't see any reversal in either of the groups. They were both trending downwards, but there was no significant difference. At 24 hours, there was significantly a better response with room temperature stored platelets compared to cold platelets with collagen. With arachidonic acid, it kind of mirrors the Verify Now, at least for the 1 and 24 hours, where we had, at least for the 1 hour, we had significantly a better response compared to cold platelets. But similar to the Verify Now here, we didn't see any response in these platelets. Now, the reversal is here plotted with the Verify Now, with the pre being the post-loading dose. So the inhibited is normalized to inhibition, or I guess the change is plotted as from inhibited. And you can see here, this was the previous study. Both groups appear to reverse the effect of aspirin, but at 24 hours, the cold group decreased. And here, this is the current data at 7-day room temperature, 14-day cold. We had some reversal with the room temperature, but no reversal seen with the cold group. So how would these platelets work if we can detect any integrin activation? What we also looked for is pre and post transfusion thrombin generation. And what we found is that in recipients of cold platelets, the peak thrombin was significantly higher compared to room temperature stored platelet recipient. Also, the time to tail, so the time from peak to end of thrombin generation, was significantly shorter, and there was a trend also for time to peak thrombin generation being shorter in the cold group. So this appears now, at least from our point of view, how these platelets, extended cold platelets, could work is by, similar to frozen platelets, generating more microparticles and being more procoagulant. And luckily, we still have samples where we can do multi-omic studies on, can try to correlate bag markers to responses in the recipients, and that being integrin responses in the room temperature stored platelets and procoagulant responses in the recipient. We also have the samples from the recipients, so we can test platelet poor plasma, see if we can still detect more thrombin generation, which would hint at more microparticles being in the recipients and causing that increase in thrombin generation. So to summarize this first part, I showed that the recovery of these platelets in vivo decreases continuously over the time of storage. The platelet GP6 levels decrease in cold platelets relative to room temperature storage, resulting in decreased responses to collagen in humans and convalescent in mice. Then extended cold stored platelets show reduced integrin activation, but appear to generate more thrombin in the recipients than room temperature stored platelets. And lastly, what we're currently working on is the role of additive solutions for storage and preservation of receptors, for example, GP6. Now, in the next part, I would like to focus on the GP1B59 complex and platelet storage. GP1B59 complex was discovered in the 80s, and it was shown that it was the primary complex to interact with VWF. And this was also shown to decrease by shedding, and that being a hallmark of the platelet storage lesion, also shown in the 80s here by Mickelson and co-workers. In a subsequent study, Wolfgang Bergmeier showed that ADAM17 mediates the shedding of GP1B alpha, and if you inhibit this shedding, you can improve in vitro aged and damaged platelets. So the way this works is that if you have exposure of VWF at the site of injury, there's an initial tethering step where platelets interact with GP1B alpha and VWF, and this is dependent on GP1B alpha. Now, it has been investigated before, and the GP1B levels decrease during storage, as I mentioned. That appear to be more so the case in cold platelets compared to room temperature storage. What Karen Hoffmeister's group from Boston showed was that the GP1B alpha is redistributed upon chilling platelets. So it's redistributed from a linear arrangement to clusters in the cold. And these clusters actually appear to increase binding to VWF, and this could help the clearance of these platelets in recipient. This was shown recently by Renhao Lee's group from Atlanta. So what we did is to perform proteomics of room temperature and cold platelets, and we washed platelets, lysed them, reduced them, alkylated them, and digested them, then analyzed them by mass spec and ran them again against a human protein database with a commonly used false discovery rate of 1%. And we were able to corroborate the previous findings that you see a decrease of 1B alpha over storage time. This seems to be more pronounced in the cold up to 7 days, but at 14 days, it's reversed and it's actually lower at room temperature storage. The same is true for GP5. So you can see a decrease over storage time with slightly higher levels at 7 days for cold platelets, and then a reduction in room temperature, especially pronounced after 14 days. And we also looked at the other parts of the complex GP1B beta, and overall no significant changes were observed. And similarly in GP9, we observed relatively stable levels. Yet at 14 day, it still turned out to be significant, but compared to 1b alpha and 5, this was definitely the degree of reduction was lower. So to further investigate this, we looked at the extracellular peptides and the cytoplasmic peptides of GP1b alpha. What we found is that the strongest reduction was observed in room temperature for the extracellular domain, hinting at a shedding event. We then performed a ratio of extracellular to cytoplasmic peptides, and this ratio was significantly lower here for room temperature storage. Stored platelets, again indicating that shedding could be the predominant mechanism for room temperature storage, at least. Now, we did this for part of a different study, but we know also that microparticle generation is more pronounced in cold platelets. So ultimately, we hypothesized that cold platelets could lose GP1b alpha via a microparticle formation. So to test this further, we wanted to use an ADAM17 TACE inhibitor that prevents shedding of GP1b alpha and could improve hemostatic function in mouse models. So we had collected apheresis platelets and tested GP1b alpha levels at baseline and microparticle formation and soluble GP1b alpha at baseline, and also performed flow chamber studies. Now, these are all still ongoing studies. I can only present partial results here for this study, unfortunately. But for the both groups were then stored with either a vehicle or inhibitor, and then the same experiments were done at 7 and 14 days to see the effect of ADAM17 inhibition. We used a TMI, which is a broad spectrum MMP and ADAM inhibitor. It's more 17 than any of the other ADAMs that it inhibits. And indeed, what we expected was true, that incubating with TMI prevented GP1b alpha shedding in room temperature storage at the extended time point. And this was not the case in cold platelets. So we're currently looking further into this, into the role of microparticles for loss of GP1b alpha, and the role how platelets, cold platelets, lose GP1b alpha. So to summarize the second part, GP1b alpha and 5 levels decrease during storage at room temperature and in the cold. Proteolysis of GP1b alpha occurs at both temperatures, but clearly at room temperature is the more predominant mechanism. And cold storage leads to more microparticle formation. And most recent results that we have from this study is that inhibition of proteolysis preserves GP1b alpha levels in room temperature stored platelets. This is not the case in cold platelets, again hinting at a different mechanism. And finally, to just focus a little bit more on the clinical part of cold platelets, we're just about to enroll, hopefully, the first patient this month. It has been incredibly difficult to set up this clinical trial, but we are starting a clinical trial with Swedish Medical Center, which is one of the major tertiary care hospitals in Seattle, where we're comparing room temperature stored platelets to cold platelets in cardiac surgery patients. This is a trial synopsis here. You can see the objective is to test the efficacy and safety of apheresis platelets stored either in the cold or at room temperature in cardiac surgery patients who are actively bleeding and who require platelet transfusion. We have plan to enroll 30 evaluable subjects, so 15 in the cold arm and 15 in the room temperature stored arm. It's an interventional randomized double-blind control trial, and we plan to use ABO-compatible apheresis platelets for this trial. This is an outline of the flow chart, so we'll address them when they are consented for surgery. We only include redo cardiac surgery patients because we know from our data that these patients are most likely to bleed. And since we have to get platelets for every enrolled subject, we want to minimize the waste if patients do not receive transfusion. So the primary endpoint, this is a pilot trial, and I hope when we're successful to perform the trial, we'll ask for more money to include more sites into this trial. But the primary endpoint at this point is recruitment and accrual feasibility of the protocol. And we also have secondary endpoints like total allogeneic blood units transfused, red cells, platelets, and plasma, and a composite of all three. Volume of chest tube output, platelet function will be done by my group, and administration of hemostatic agents as well. We also look for the need for surgical re-exploration due to bleeding within 24 hours, the new infection of sepsis within 3 days, new thrombosis. Our initial plan was to follow up to 3 days, but the DSMB members thought this was too short, and we had to include data up to 7 days. And there are good publications to support that change. Then we include liver injury. Now, compared to I'm not saying this is, it still seems a little bit far-fetched, but we know that while room temperature stored platelets are predominantly cleared by the spleen, cold platelets are predominantly cleared by the liver. So I don't think there's any good data out there to support that this could be harmful to the liver or anything. But the question is really, do we detect any change, even if somebody receives multiple units of cold platelets? Then total number of adverse events, hospital duration, post-surgical ICU duration, mechanical ventilation, and 28-day survival as well. We also have some exploratory endpoints where we use the stasis assay, the assay that I mentioned before, that uses a block and post assay. And this assay has been published on before. The advantage is there's an automated bench system. It's sensitive to platelet function inhibitors. This has been published by the group who invented this assay here in 2019. And in the same publication, they show that it predicts need for transfusion in trauma patients, unlike aggregometry or ROTEM. And a second exploratory assay, we look into the recovery of function in vivo. Now, the way we want to do that is by looking into the differences in HLA. And I think this has been done by, for example, groups in sanguine, predominantly in Europe, because they don't have the radiolabeling capability. But in this case, I think it makes sense because we are able to test the platelets that are going to be transfused before, and we have the patient's HLA type also. So we could identify the transfused platelet population and the endogenous platelet population, and then identify a recovery of function of the transfused platelet population in the recipient. So this is a little bit of a cartoon where we test the HLA type of the donor and the recipient, and then focus on platelet function of the endogenous versus transfused population. And we try to stimulate platelets and look how well they respond, platelet surface markers, and phosphatidylserine positivity, for example. Now, with that, I would like to close and acknowledge everybody from my group who worked on this, on all of these different projects. I also showed data in collaboration with Jose Lopez's group, the FU lab, the mass spec core here at Bloodworks Northwest, Nate Snydecki's group at the University of Washington, the Department of Engineering, and Jane Fei-Dong's group here at Bloodworks Northwest, and all the funding agencies as well. And thank you very much for your attention.
Thank you very much, Moritz. We have people can put questions in the chat, or you can raise your hand. We do have one question here from Dr. Brugnara, who says, the MD Anderson group has published some initial data on freeze-dried stabilized platelets. What's your opinion of this approach?
Yeah, That's another good point. Yeah, so it's not only frozen platelets, but lyophilized platelets, so freeze-dried platelets as well. I think I'm not really aware of any data that they are better or more beneficial than frozen platelets themselves. I think there may be advantages in the preparation that they are. You'd only need to reconstitute them with saline, and then they're basically ready to use. You don't have to undergo the thawing process, I guess. So that may be one of the major advantages of these of the lyophilized platelets. But otherwise, functionally, I'm not aware of a head-to-head comparison where they performed better than frozen platelets but I think the mechanism likely is going to be the same, that it provides procoagulant surface and aids hemostasis that way.
I see Dr. Bucel has his hand up. Jim?
Hey, it's a really interesting talk. I have a comment and a question. To what Dr. Brugnara said about the lyophilized platelets, and big advantage that maybe is obvious, but I don't think you mentioned, is that they can be stored for up to 3 years even.
Yeah.
And that's a huge issue, I think. What I wanted to ask you is, some of your platelet function assays, let's call it, don't really reflect the possible, let's call it, beneficial effects of microparticles. How important do you think that is, and how do you think you should handle that best in the future?
Yeah, I agree. This is a very, I was, I guess, based on my training, was mostly focusing on integrin activation, I guess, and p-selectin exposure, and focused on that. But as it turns out, in the last study, that was the first time we included the thrombin generation assay, and that was really the one time we detected a benefit, I guess, of cold platelets compared to room temperature stored platelets. So I completely agree. I think this is definitely at least that's what it looks like for me right now. That's the way these platelets are going to work in the recipient, is by microparticle generation. So I think in order to address this, we still have the plasma samples of the recipient. So we wanted to test by flow cytometry. We now have the Cytoflex, which apparently gives us the best resolution up to very, very, so we can actually differentiate different sizes of microparticles with this new instrument. So we will look into platelet poor plasma samples of the recipients and look into microparticle populations. And I think, I'm not a microparticle expert, but the fact that we see more thrombin generation and overall a shorter time to peak and time to tail looks, at least to me, that the reason for that is or could be microparticles. So yeah, we want to demonstrate microparticles, and if we still, I guess, with platelet poor plasma, we could also expose them to the thrombin generation assay, and then see if we still get more thrombin generation in the cold recipient group versus the room temperature recipient group, which would also prove then that it is the microparticles that help with more thrombin generation.
Thank you. So we have another question in the chat box from Dr. Nantel, who would like to know, does the decision on storage temperature, whether it's at 4 degrees or 22 degrees, need to be made at the time of collection? Or can 22 degrees stored platelets be converted to 4-degree platelets after 3 or 4 days?
Yeah, that's a great question. I received a lot of questions about this from other groups. And so for our trial, and basically for all of our data, we put them in the fridge within 2 hours maybe. So really, very, very quickly. But this is obviously not practical for a blood center. So for the human trial, we allow up to 4 hours. And but I believe there was a study that looked also for much prolonged period of time, for example, 24 hours. And I think they were still turned out to be fine. I don't think, though, if you wait too long, I don't know, 2, 3, 4 days, how that would affect the quality of the cold platelets, and you would still get that pre-activation that you get with cold platelet storage. I'm not sure about that.
Yeah, I think there was a paper that was published by our colleagues in Australia showing that at least with the in vitro markers they were looking at, that platelets looked quite similar put into the refrigerator at day 4 as they did if you put them in on day 1.
Okay. Oh, is that Denise Marks' group, right?
Yeah. I think Lacey Johnson's probably the first author.
Mm-hmm.
Okay, we've got another question here from Dr. Carter, who would like to know, should you be using markers of systemic thrombogenic effects by measuring something like fibronopeptide A?
Yeah, it's a good point. I think we have not done it clear, but I think we should definitely include markers like that in the future for sure. Yeah, like I said, I think based on my training, I was too much focused on just the platelet biology aspect, but other markers would definitely be helpful as well.
Yeah, and then Dr. Conway would like to know what happens to platelet function over time in cold stored whole blood.
That's a good question. The cold stored whole blood, I've never used cold stored whole blood. I think from what I remember from Cheryl's data actually, is that they actually tend to be better preserved than if you separate them and store them in plasma. There was a study that I think she published 5 or 6 years ago together with Dr. Kapp's group.
Yeah.
So, but then, yeah, you basically stuck with transfusing whole blood, I would say. Otherwise, you would have to separate them after storage, which could be tricky with the stored platelets. At least in our hands, if we store platelets and then try to wash them, for example, or further manipulate them, it gets really, really difficult to get them back into suspension.
Yeah, and I think if you look at all the literature on platelets in whole blood, you tend to build larger aggregates the longer you leave the whole blood in the refrigerator. Mm-hmm. I guess that's not too surprising given what Karen Hoffmeister's shown.
Yeah, true.
Okay, Dr. Prysdale would like to know, did platelet proteomics analysis that you did include the microparticles? And if so, is it possible to separate the microparticle proteomics from the whole platelet proteomics?
Yes, I think it is possible. And it's important. Yeah, it's a great question. It's important to point out that we only included the pellet for this analysis. So we did not include any supernatant, no microparticles. But it's definitely we want to do something we want to do in the future to do the same analysis on microparticles as well. But yeah, for this study that I showed the data for was just a platelet pellet, so no supernatant.
Yeah, Moritz, have you looked at the metabolomic profile of your cold platelets?
Yes, yeah, we have, but it wasn't really so striking. And then I think you have published on proteomics, right? But then I think the Alessandro's group together with I forgot his name, Spinelli, Dr. Spinella. Yeah, he published on metabolomics of cold platelets already, right? So we didn't, and I don't remember there were like extremely exciting findings, at least so far. I think what I'm interested most now is to find maybe like predictors for procoagulant function and that's what Jiayun Fu is currently working on here at Bloodworks with the data that I've just shown you, to identify predictors of procoagulant function, for example, or integrin activation in room temperature stored platelets. But I don't know. Yeah, that's really all I can say. Unfortunately, I know you're also very interested in the omics.
We had a collaboration with Angelo D'Alessandro looking at the metabolomic profile of cold stored platelets, and I don't know if I want to put my graduate student on the spot here, but Wayne, maybe you want to summarize what you found.
Yeah, I don't mind at all. Hi, Dr. Stolla. So what we found in that study was a lot of the pathway related to the glutathione was really important in terms of platelet function. So we did a correlation with our in vitro study. We did rotom and agribometry data with the metabolomics and targeted metabolomics approach. And one of the pathways that popped out to us was the pentose phosphate pathway, where we think that pathway was really significantly elevated in the room temperature setting, platelet setting. We saw really low levels of glutathione in its function against oxidative stress. So our next step to approach that would be to look at the pentose phosphate pathway and looking at inhibitor studies with that. So that's a really quick summary, but we found other significant pathways as well, but we're sort of doing that step by step. This is one of the top priorities for our study right now.
This was a comparison with room temperature storage?
Yeah, so what we did was we took the standard issue of our bulky cold platelets from Canadian Blood Services, and we stored them either in the room temperature setting or in the cold at 4 degrees. And then we've collected sample data points on various days, and we send those out to do metabolomics and do in vitro studies in the lab.
Okay, nice. And you saw increased oxidative stress. Did I understand this correctly?
Yeah, that's correct. And we saw the metabolites of the pentose phosphate pathway were elevated in the room as well.
Very cool. Yeah. Yeah, I was trying to convince Dr. Devine to have maybe some shared lab meetings once in a while or something. I think it would be incredibly interesting to.
Yeah, would be for sure. We'll happily do that.
Great.
So do we have any other questions? I think we've addressed all the questions that were in the chat. Does anyone want to ask anything else? If not, Moritz, thank you very much. It was a really interesting presentation, and I can guarantee you we will be in touch soon about dissecting all our data together.
That would be great. Thank you so much for giving me the opportunity.
Our pleasure. Okay.